Yeah, we can see it.
Okay. Okay, hi everyone. Welcome to STEMEY molecular biology class. Today we're going to go over photosynthesis. So first, the overview of photosynthesis. The equation for photosynthesis is 6 molecules of H2O, also known as water, combined with 6 molecules of CO2, also known as carbon dioxide, that creates C6H6O12, which is glucose, and 6 molecules of oxygen. So basically what this means is that it converts light energy to chemical energy. And photosynthesis is used in plants, algae, and cyanobacteria. And in plants, it occurs in the chloroplast. So you can see a picture of the chloroplast on the top right. And there are 2 steps to photosynthesis. The first is the LDR, which stands for light-dependent reaction, and then it goes through the Calvin cycle.
Okay, so for the structure of the chloroplasts have a double membrane, and inside the double membrane there's the stroma, which is a dense fluid, and they also have thylakoids, which are sacs that also have membranes that separate the lumen from the stroma, and there's the chlorophyll, which is the pigment that's found in thylakoid membranes, and this is what helps absorb light energy.
Okay, so as I mentioned earlier, the first part of photosynthesis is the light-dependent reaction. So the light-dependent reaction occurs in the thylakoid, and it converts light energy to chemical energy, which is in the form of ATP and NADPH. And so the steps of the light-dependent reaction. First, the absorption of light excites the electrons that travel down to the electron transport chain. And then water is split into oxygen and a hydrogen ion and electrons. And then the oxygen is released and the hydrogen ions build up gradient in lumen. And then NADP plus is reduced to NADPH, and then the diffusion of the hydrogen ion into the stroma powers ATP synthase via chemiosmosis.
Okay, so next is the Calvin cycle. So the Calvin cycle occurs in the stroma, and it uses ATP and NADPH to make sugar from carbon dioxide. So the steps of the Calvin cycle are carbon fixation, where carbon dioxide combines with an organic compound called RUBP. And then the next step is reduction, where electrons are added using energy from ATP and NADPH to make G3P, which is a sugar that makes glucose. And the next step is regeneration, where some of the sugars that make up glucose, the G3P, are recycled to regenerate RUBP. And this releases NADP plus and ATP to be used in the Calvin cycle.
Okay, so there are also some alternative methods of carbon fixation. So first is photorespiration, and this is when O2 is fixed instead of CO2, and it doesn't produce ATP or sugar. And then C4 plants minimize photorespiration by fixing CO2 into a 4-carbon compound that can release CO2 to keep carbon dioxide high in bundle sheath cells. And then CAM plants store CO2 as organic acids that can release carbon dioxide during the day. And this reduces water loss because it closes the stomata during the day. We're going to skip our activity. You have no homework. And thank you for coming.